Ada Yonath, Venki Ramakrishnan, Thomas Steitz, welcome to Stockholm. You three are united by your studies of the ribosome. And so this is the molecular machine which lies at the end of what Crick called the central dogma of molecular biology. DNA makes RNA, makes protein, and the ribosome is what translates RNA's code into protein. And deciphering its structure and workings is a lifetime's work. So I wanted to start just by asking each of you what it is that appeals about taking on a task which is so enormous that you really do have to spend a lifetime sorting this puzzle out. So if I could start with you.
Well, I started about 30 years ago to get the structural basis of Crick's central dogma. DNA makes RNA makes protein, as you just said. And we started with DNA polymerases and then looked at RNA polymerases and some of the proteins that are involved in for example, putting amino acids on tRNAs. And then finally, in the mid-90s, we got to the point where the ribosome was the really important target, and Ada Yonath had already shown that, in fact, crystals could be grown, and she had some crystals that diffracted pretty well. So it seemed like that was the next mountain we wanted to climb, and indeed it was a very exciting adventure.
Mm-hmm. It's an adventure which is, but it is sort of absorbing all of one's time and passion. Does it not sometimes seem like an adventure that's too large to undertake?
It didn't seem too large, although it wasn't so clear how and when we were going to get there. It was clearly a challenge. But, indeed, in fact, it was took less time than many of the problems in the lab. Not that we expected that to be the case.
So I got into ribosomes almost by accident. I was switching from physics to biology, and I had written to a number of universities asking whether I could join their graduate program to do a second PhD in biology. And at the time, Tom won't remember this, but two people from Yale wrote to me. The chairman of Yale wrote to me and said, "We won't take you as a graduate student, but I've circulated your letter to faculty members, and some of them may want to take you on as a postdoc." And two people wrote to me, Don Engelman and Tom here, asking if I would be interested in doing a postdoc. And I wasn't ready for it because I felt I needed a broader background in biology. So I went to the University of California at San Diego to study biology. And then after a couple of years, I realized I didn't actually need a second PhD. And I then wrote to Don again. And at the time, Don Engelman was collaborating with Peter Moore on the effort to locate all the individual proteins in the small ribosomal subunit. And so Peter Moore met me at some meeting and then kindly accepted me in his lab. And so that's how I got into ribosomes, and that was in 1978. And then when I had my own lab, we started working on pieces of the ribosome. And then in about the mid-90s, I had some ideas of both how to try to get better crystals of the small ribosomal subunit, for which there weren't very good crystals at the time, as well as perhaps what to do if I actually got crystals. And so with these sort of ideas in mind, we started tackling this problem of the whole ribosomal subunit, which I thought of as a sort of stepping stone towards looking at the entire ribosome.
Mm-hmm. But again, this idea of having to take stepping stones to get to the eventual goal, it does, from the outside, look almost impossibly daunting that one begins so small.
Not if you look at it one step at a time. I think, we were looking at individual ribosomal proteins, then we did a protein RNA complex in the ribosome, then we did an initiation factor. So it's sort of each time, I think the complexity increased a little bit, but then I think I agree it was a big jump to try to sort of work on the whole ribosomal subunit, which of course people like Ada had been doing for quite some time by then.
Mm-hmm. So you sort of began the Herculean task by attempting to get a crystal of the ribosome from which you could derive a structure when people really were saying this couldn't be done. What turned you to the subject in the first place?
So to start with, I think that people began before me. There were some that tried to produce crystals of the ribosome in order to determine the structure. So I knew about some attempts, some even serious attempts around the world. But what my way in getting into it was very similar to what Tom told. Anything that has to shed some light on protein biosynthesis was of interest for me, because the process attracted my curiosity so strongly that I was ready to do almost everything. So I started with the initiation factors, and when I needed some more material, I don't want to go into all the detail, I got into the idea that maybe ribosomes can be crystallized by reading not directly related journals.
I see from your lecture title that story is going to come up in your Nobel lecture.
I think so. But in the beginning, I thought, as I said earlier, any progress in this very complicated and very challenging process would be of interest, the translation of the genetic code. And I thought that even if I step just one little step forward with not so good crystals, with better crystals, with this type of crystals, or crystals that can be only viewed by electron microscopy, whatever can be done that will increase our knowledge and our understanding of it would be, for me, satisfactory, almost completely, but not fully. So curiosity is what drove me. And we had to go through obstacle after obstacle, which took about 15 years until we showed the feasibility, as Tom said earlier, and also Venki. And eventually we got there. But I think my driving force was curiosity, the wish to understand this process. And I didn't think about this is too big, too large, or whatever. I was ready to go more or less step by step.
Did other people around you believe in you? Do you think they believed that you were going to...
Very few. But the few that believed were very good and quite supportive. Very few. The person I collaborated with was the director of the Max Planck in Berlin, Heinz Günther-Wittmann, who prepared the materials and prepared the ribosomes and had the manpower to produce what I needed to try. Sir John Kendrew, who was the head of EMBO at that time, and EMBEL, made it possible for me to go to synchrotrons and try to measure, although the synchrotron radiation people not always wanted me there. They said, oh, you don't have any result, you go. But he said a project like this shouldn't be stopped. Luckily, he was also on the scientific advisory committee of the Weizmann Institute where I worked, and once he said this sentence, the Weizmann supported me for a long time going on. Aaron Klug from MRC, whom I visited by accident because I came to see somebody else, and asked me what I'm doing there and looked at my initial results, thought and said, you know, leave it here, maybe we can help you. So I knew that I have something, and this was back in the beginning of the 80s. And Ken Hobbs, who said, even if there are no ways to continue after crystals, they will be found. Just go on. And then on the way there was a lot of up and downs, but
Did you welcome the advent of other people in the field? Because as success began to come, more people joined the field. Was that something that you saw as a positive thing, or was it worrying that suddenly an area that you'd had to yourself
was... So I have two answers. First, a general answer. I think that for science it's best that more and more people are interested in basic problems because they have different views, they may have different techniques, they may look at things with different attitudes. So for science, at the end, this is very good. For me, it was wonderful. The first time I heard that Tom has crystals, this was the first time in my life, ribosome life, that people didn't tell me, oh, you're dreaming, or you don't really have what you have, or whatever, all the attitude I had earlier. So this was, and Tom told me it was easy to repeat, so it was good.
I think it's true that all scientists stand on the shoulders of those who went before, and so and it doesn't have to be very far before, and that is we help each other. I mean, obviously we benefited from Ada's early work. I think we did some things that might not have helped some of the others in their approaches, or at least seeing that it can be done. So I think this is a very helpful thing. You don't want to do science in a closet. It's a very, first of all, it's not any fun. And second of all, it's not as productive and as exciting.
I also want to say that in the mid-90s, when both Tom and I entered the field, it was still far from clear that there was a solution in sight, okay? And in fact, both of us had to do, had our own approaches of how to get the job done. And in fact, we've, used some of Tom's ideas for getting low-resolution phases. So I think it isn't quite right to sort of say, well, all of the groundwork was laid and then the, others sort of joined in. I think it was far from a foregone conclusion, and so much so that I actually I felt that I needed to leave my professorship in the US to seek the rather stable funding environment of the LMB in Cambridge, because I had no idea whether it would take 5 years or 10 years or longer. So.
So if I can still add, because I didn't finish your earlier question, there were two more people that did believe and did help, and I would like to mention them. It's Hakon Hope from Davis, with whom we found a way to eliminate or at least minimize crystal damage at the X-rays, a method that became routine right away afterwards, cryobiocrystallography. And he came in a very critical minute when I thought 86, 87, when I thought that the whole thing is gone. And Michael Pope from Georgetown, who provided us with the clusters, with the polytungsten clusters that were used for phasing by us, by Tom, and by Venki, at least at low resolution. In our case, even at high resolution. So I think once I talked about those people, I would like to mention them, and Michael Sella, who was the president of the Weizmann Institute, that made clear that I can continue.
I might return to the topic of competition again in a little while, but you mentioned the LMB, and so you still work at the MRC Laboratory of Molecular Biology in Cambridge, and you were a postdoc, I think.
I was a postdoc there, and I did a sabbatical there as well, for half a year.
So it's one of those famous places that people talk about and has generated, for instance, a large number of Nobel laureates. What would you say was its defining characteristic that makes it successful?
I think there are several of those. One is that it has core funding from the Medical Research Council, and the MRC has always understood that the LMB would be tackling hard fundamental problems and not ones where you have a quick payoff and lots of papers and so on. So that's one thing. The other thing is that all facilities at the LMB are shared, and so there's a great spirit of collegiality there. Anyone from a senior scientist to an incoming PhD student has exactly the same rights to use X-ray equipment or any central facility. So that kind of interactive environment also fosters exchange of ideas and so on. And then it has a tradition of being self-critical. People are always asking each other, why are you doing this? And that's a very powerful question. If the answer is, oh, because I want to get my next paper published, it looks a bit lame. And you have to really think about, why is this thing that I'm struggling with important? And what, even if I succeeded, would it actually tell me anything? So I think that's an attitude that it tries to cultivate. So there are several aspects.
So there's a sort of self-policing going on that everybody's asking, not policing, it's the wrong word, but everybody's asking each other, what is your bold question?
Yeah, no, there's a real culture of not spending your time on what might be called sort of mundane or trivial problems. I think that's true. And I think that's also made possible by the way the LMB is funded and by the sort of organization of the LMB. So the three aspects actually are all interlinked.
I found as a postdoc, it was the most impressive environment because of the way people interacted with each other. I always had lunch, coffee in the morning, lunch at noon, or actually 1 o'clock, and then tea in the afternoon. There's so much so that I always wonder, well, how does anybody get anything done? But it's exactly what Venki just said. People would only talk about science, not what they saw on TV last night or the movie they saw. And they would always bring up what they were doing and why they were doing it. And you might happen to be sitting next to Francis Crick, and you might say, well, I wonder why you're doing that, or had you thought of the following way of solving this problem? And and within just a few months, we knew everybody in the whole laboratory. It's a fabulous environment for learning and for doing creative thinking. And I think that all these other points, like the funding and that, very important. But to me, as a postdoc, the most exciting thing was the culture of how you do science and how you think about science and how you interact with other scientists.
And do the senior scientists still go to tea at the LMB?
Oh yeah, no, that's a very important part of the culture. And I remember I went there on sabbatical in 1991, and I'd arrived the first day, and John Finch, who works closely with Aaron Klug, was my official host. And I came into the lab at 8:30, and about 10 o'clock, he came and said, would you like to go for coffee? And I thought, no, I've only been here, an hour and a half, and, so I said, well, not really. And someone looked at me and looked at John and said, he hasn't learned our ways yet. And, then I realized that actually this, these periodic interruptions in your day, they're actually, you tend to get less tired as well because, you're doing something, and the mind, I think, can only focus for a couple of hours at a time before some sort of mental fatigue sets in. So it's a natural way of breaking it up, and then you talk to other people, and it's certainly a very important point.
Mm-hmm. We shouldn't really leave out the Weizmann, I suppose, when we're talking about this. The Weizmann is a very successful institute as well. Do they try and foster similar cultures there?
I think the Weizmann is larger than LMB, so this type of interaction, everybody sees everybody all the time, or almost all the time, or 3 times a day, is just impossible. Yeah. There are hundreds of people working there. But the Weizmann does try to single out people and projects that have very high importance, very significant, and let them work even if success is not shown just across the border. Not always. The funding is not so simple, although there are some ways to make it better, like creating centers. I was very lucky to have a center like this of structural biology, fostered by Helen Kimmel from New York. Mm. But they do try no matter how the funding comes, to let people go above and beyond what is called average or trivial or sure or no risk. They do try to let things like this happening if they have a feeling that it's important and there is a chance that it will also mature. Some chance.
I should point out also that I've been supported for the last 25 years or so by Howard Hughes Medical Institute, and in some ways it's like the LMB in that it's a more stable funding organization than the NIH, which every 5 years you have to renew. And I think they've been very helpful. And there was a structural biology consortium at Yale, about a half dozen or so investigators, and we tried to emulate the LMB environment and having lunch and interacting. So I think they're money and culture.
Mm-hmm. I mean, people constantly talk about the fact that the availability of long-term money with few strings attached is becoming scarcer. And a project such as this, which has obviously taken such an investment in basic research where one doesn't really know when one's going to get to the end, is a great example of why it's needed. Do you fear for the future of this sort of bold approach outside of places like the LMB?
Well, I think even in Britain, there are movements to, constantly sort of evaluate work based on its impact, etc. And so there is a danger, but I think good science administrators understand the value of basic science. I mean, that's true in the US and it's true in England as well. I don't know about Israel. But there is a danger that because a lot of science gets funded by taxpayer money, that the taxpayer needs to know, why should a truck driver's taxes go to support particular kind of research. I think scientists need to be able to explain and make the case for basic research. It's not a very difficult case to make because, so many great inventions and discoveries that have benefited humanity have come out of basic research. Mm-hmm. And if you actually put a value on the investment and the return, you'll find that basic research is actually very good value for money. And so the good administrators are aware of it. The problem is sometimes you get people with more short-term visions controlling things, and then it's not so good.
So if I may add,
please.
A large part of my work on the ribosome was also made in parallel to the Weizmann at the Max Planck Society in Germany that even set up a unit for me near the synchrotron in Hamburg in Daisy. In the past, Max Planck was like a paradise from point of view of funding. There was hardly any problem once there was a decision, this is a project or this is even more like this, a person that can run a department called Abteilung. Recently, even there, I think, as far as I felt and as far as I know, I think that it's becoming more and more like what Venki just described. Taxpayers want to know, there is constant evaluation. Some people understand the importance of basic science and some do not. And even in places like this, like Central Europe, and where it was known that money is given almost with no strings, it's not so anymore. So I benefited from it for some time, but I also saw how it changes.
So the structures all came together in a rather sort of marvelous way in 2000, and suddenly there was a high-resolution structure available of the large subunit and the small subunit, and indeed of the whole ribosome. And that's been constantly improving since. At that stage, just getting back to the competition idea, at that stage when things were really coming together, was that an anxious time to see whether one could get into press first with one's findings?
Oh, I have to say, I think it was a very anxious time, and we were all very tense. And there was a period I particularly remember because we got to 5.5 angstroms without any problems, and that was published in '99. But to go from 5.5 to 3 angstroms, which is what we needed to get the high resolution structure, for us was rather difficult because we ran into a number of technical problems which had to do with the behavior of the crystals. And I would keep getting reports, you know, that the Yale group was steadily sort of advancing. And, you know, so it was quite a depressing time, actually, around December of '99. And luckily, in the end, you know, we were able to sort of pull it off. But I think, to be perfectly honest, it was a very, very tense time.
Oh, we thought Venki would be much faster because it's such a small subunit that he had. So, wow. So,
I once joked to Tom, it isn't fair that you guys, you know, got in a bit ahead of us because it would be like hemoglobin being solved before myoglobin, you know.
Well, that's... But you see, what held us up is we had more atoms to fit in, because we had a high-resolution map in December, I think it was about December, early January of 2000, December of '99 or January of 2000. But there were 100,000 atoms that had to be fitted into the map, and this takes a while. You probably only had 50,000 to fit in, so
that's about right.
Half as long, so.
Yeah. But crystals which were much worse, I found.
Our crystals were good, I guess I don't worry. I should worry about these things more, but I don't. And so we were just worrying about just getting there, and I knew there were other folks out there, but we thought we could do all right. We just pressed on.
There was also another element, which is, you know, I deliberately chose the 30S because at the time I started, there were good crystals of the 50S, but there weren't any good crystals of the 30S. And so I thought, when Tom started working on the 50S, that he and Ada would be competing and I would be quietly having a good time with the 30S. And then I found out, you know, barely a year after I started, that Ada had her own good crystals of the 30S. And so instead of Tom and Ada sort of duking it out, it was, you know, a head-to-head race between me and Ada, which was not the position I wanted to be in. So.
So if I may add something. First of all, it's a very good question because it's the first time I heard that Venki was under pressure. I thought that Venki was quiet and went step by step. He was so quiet that we didn't even know that he works on it for, I think, 2 years, more or less. And we had the 2 subunits at that time. The same one that Venki had, but we got it differently. And a different one that Tom had, but a few bacteria. So we had to go both ways. And somehow I felt that in my group, the feeling of race was not the most important thing. It was not so important to be a week earlier or a week later as it was important to see the structure. I felt so. Maybe I'm too easy on my people, but we were also geographically a bit further away. But it's very good that you asked him.
No, but I think all scientists are fundamentally curious and are driven primarily by, you know, wanting to see the result. But we're also human, and we'd like to see it before other people.
And you don't want to see it in the next issue of Nature.
Exactly.
While you're still interpreting your map.
Yeah. So I think it's honest to say that most scientists like to see the result, but they'd rather see it first, you know, before someone else
maybe it's right for most.
Yeah.
So, if one tries to describe how the ribosome works with our current picture of it, if you sort of, I don't know whether one wants to use one's hands or whatever, but how would you describe this molecular machine as you see it when you picture it working? I mean, broadly, the large subunit is building proteins and the small subunit is checking their integrity, I gather.
No, it actually it has to check whether the right amino acid has been brought in before the amino acid gets added on, because otherwise it's too late. So.
We call the small subunit the clever one, because it has to find the right piece of the messenger RNA to be translated and bind to it, or bind it to whatever you want to call it, and then it has to select for the right amino acids. That's what Venki did now and said now, and also prove it. In the beginning, before the large subunit, all what it does is they put them together. It's not as clever as the small one. But then we found out that also putting together is not as simple as it is to say it.
But at what sort of speed is this molecular machine working, and should one think of it as a machine, or?
Oh, definitely.
Yeah.
It's a machine. It uses energy and it moves, so it has all the properties.
It reads instructions. It gets instructions. It comes out with a product. So it's a machine.
It's like translating these punch tapes that used to, nobody knows that anymore, but we used to have paper tapes that we would use for running our x-ray machines. And you would punch the tape and then you could read that out to get the intensities. Well, that's what the ribosome is doing. It's reading out this paper tape, the messenger RNA, to say which tRNA containing which amino acid should be delivered. And that's what the small subunit is doing, is making sure the right tRNA is delivered to the right message. And then the large subunit, once the amino acid has been delivered, is attaching it to the growing peptide, and that's the function of the large subunit.
It does more than this. After it attaches, it also protects it, because this little protein starts from little until it grows up. It protects it. So the first one is selecting, the second one is protecting.
It has this sort of pouch.
It has a tunnel to do that. But
So one thing is that you asked, how fast is it? And it synthesizes 20 to 30 residues per second with an error rate of less than 1 in 10,000. Now, one reason to mention it is every time, the Nobel Committee gives a chemistry award to something that's in molecular biology, let's say, there's always a hue and cry from hardcore chemists saying, well, these people aren't real chemists, and they've gotten our prize again. And the fact is, there is no peptide synthesizer today that can come even close to making 20 to 30 residues per second with that kind of accuracy. And so, well, I may not personally be a chemist, but the ribosome does some amazing chemistry to get it done.
So Venki studied...
And you're trying to understand the chemistry, and our view is, in order to understand the chemistry that the ribosome is doing, one must, as one piece of information, have the structural of every state in the process, and that's what we're doing.
So Venki studied physics and biology. I studied chemistry. Yeah. And I was a good student. I was considered as one that can do synthesis. I can't do 15 or even 20, even not 10 in a minute, a second. I can't do it in a week.
So there's no question the ribosome is a good chemist as well. It can do this. But do we on this question of whether this is a chemistry prize or another prize, do we understand the ribosome at a chemical level? Do we understand the way it does this chemistry?
Much more so after these structures.
Well, I think it wasn't understood.
At least we know what to ask.
No, I think the mechanism by which the ribosome is a ribozyme is now understood. It was proposed by Francis Crick in 1968 that the ribosome was probably going to must have been a ribozyme, because how could you have a protein synthesize the first protein before you can't have a protein machine to synthesize protein, the chicken and the egg problem. So it had to be an RNA. And so what the structure showed, both for the small subunit and for the large subunit, is indeed, it is a ribozyme. That is, the critical functions are executed by RNA. But then the chemical question is, how can RNA be a catalyst? It's actually not most chemists' choice for a general ester or general base to use bases and sugars to do this. And so how does that happen? And I think the combination of the structural studies that we've done, plus some mutational and kinetic studies that others have done, have now led to an understanding what is the mechanism whereby the ribosome catalyzes peptide bond formation, and lowering activation barriers, chemist's language, increasing the rate of catalysis, lowering the entropy of substrate orientation, all chemistry phases. So I think we're doing chemistry myself.
So this is actually almost fully clear, what Tom just said now. But how the whole thing goes on and how it starts and how it finishes and how the cell and the ribosome interacts, this is still not fully understood. And there are questions to ask. And I think the structures directed new type of questions that hopefully will answer all what I meant and probably much more than that.
So one consequence of the structures was that it gave an insight into how one might design new antibiotics. That antibiotics, I gather about 50% of antibiotics target ribosomes. So having a structure of the ribosome should allow you to design new antibiotics. And you indeed have started a company, Ribex, which tries to do exactly that. Is it the case that we now have new antibiotics in clinical testing which have come directly from a knowledge of the structure?
Yes.
Yes.
Yes. Yeah. So in 2001, Peter Moore and myself and Bill Jorgensen started a company, Ribex, because we realized that the structures of the antibiotic complex with a large ribosomal subunit could be a useful start for designing new antibiotics. And they've been working on it now over the subsequent years. And they have one compound that has successfully completed phase 2 clinical trials that's effective against all methicillin-resistant Staph aureus. And they have several others in the pipeline. And their whole strategy is to take a portion of one compound that's bound and tie it to a portion of another compound that's bound nearby. So it turns out that the ribosome has many different families of antibiotics that bind nearby in the peptidyltransferase region, center region. And so by taking different components and making a hybrid molecule, you can now make compounds that are very effective against resistance strains. And so I'm extraordinarily optimistic that it's not only going to lead to one compound, it's going to lead to many compounds. In fact, they are now using the ribosome structure to design de novo compounds that are now showing activity against gram-negative bacteria like E. coli. And there are very, very few compounds out there that are active against gram-negative bacteria. It's a major problem. And they have several families that they are now developing that are lined up to go into phase 1 clinical trials. So I really think this is going to be another machine generating not just one antibiotic, but many. And many people ask me, they say, well, is it possible to design an antibiotic that will now always be useful and won't generate any resistance? And I always say that evolution trumps intelligent design. So however intelligently you design your antibiotic, evolution will catch up. And so what you have to do is continue to evolve your design. And I think now this is a machine that allows the evolution of the design.
And it's a desperate need as well, isn't it?
A desperate need. And in fact, we've just now, we haven't published it yet, but we've made a complex between the 70S ribosome and capreomycin and viomycin. And these are, capreomycin is one of the major antibiotics that targets tuberculosis. And there's some strains emerging, particularly in South Africa, that are called XDR, and these strains are resistant to all of the antibiotics that target TB, no matter what the target molecule is. And so they're very, very dangerous and exploding. And what we find is that this binding site for capreomycin on the ribosome is right next to two binding sites for antibiotics that Venki saw for paromycin and hygromycin. And so, I hope I got the names right. and it looks again like here's another opportunity to make a combo for new antibiotics. Now the problem with this is, of course, the people who get TB these days are very poor, and so drug companies are a little reluctant to get into this. But I think this could be a problem that will spread worldwide if we're not careful. So I'm hoping that this potential solution to these problems that can be generated by use of the ribosome structure will be very helpful.
I should say one thing about the last comment Tom made, which is, so I should say that I'm a consultant for Tom's, the company Tom founded. I'm also on their advisory board. But so I've seen it firsthand how expensive it is to actually get something through to market. It's tremendously expensive. And so it is reasonable for pharmaceutical companies to want a return on their investment. But at the same time, if large populations in the third world are deprived of these medicines, it's a serious problem. And I think, things like the orphan drug program are absolutely essential. And perhaps multiple pricing in different markets is another way, because, a Western pharmaceutical company is not going to get much money from poor countries in Africa or Asia anyway. And so they as well license it at very low prices there, provided, of course, there's stringent controls against, smuggling. But that'll be a problem whether you license it or not. And so I think this is something that people do need to think about.
So new models are needed in order to get drugs out there.
Yeah.
So if I'm allowed to add something from the basic science, from the basic research point of view, less from the company's point of view, there are ways to improve and to understand better antibiotics and consequently also resistance. For instance, we found in the literature recently 2 compounds that can be synergetic. They are not strong, and we think now that we can make them strong. Whereas very, very similar one is competing. And on this hardly seen differences, we are now building, or can build a strategy of making antibiotics that are powerful and probably less prone to resistance, or resistance will happen, but probably slower and not so aggressively if we play on this little difference that can change between synergism and competition and make it more synergetic. And it's we found it in natural products that are not in use. So there are things like what Tom and Venki described, making new ones, and there are some that are using what the nature gives with improvements, need improvements, chemical improvements, but are possible to be made. So not that we have a company for it, but we have an idea.
Yeah, but there are various strands to explore. Yes, it's nice because it was a basic science story which now has a very much needed application, but I suppose it's worth stating that it all came from basic research without the thought of that.
And what's more, I think it's true with Venki, it's certainly true in my case. I never started working on the ribosome because I thought of its practical consequence. In fact, I should have known that the ribosome was a major target of antibiotics, but I didn't really. It was only after we started that I realized this and we started working on it. And I must say, one of my concerns about what I see with funding agencies increasingly is this desire to have translational research. Translational research as applied to the ribosome is fine, but I think translational research in the sense that you want to target your research on doing something practical, I think it's the wrong way around. I think you want to understand the world and then use that information to solve the problems that it enables.
It is in universities and research institutes. Maybe companies have to think differently.
They probably do, but yeah, that seems like.
But basic science was my motivation, and I think also Venki's, in terms of understanding the ribosome or understanding translation of the genetic code, not translation.
I think funding translational research as such is okay, but what's happening is there's an increasing trend to ask basic research proposals to justify, themselves on the basis of potential implications. And I think it's very, very hard to predict, what sort of implication your basic problem will have, 10, 20 years down the line. So,
as Yogi Berra said, nothing's harder to predict than the future. And so, who knows?
On that seems like a good point to stop with, although it's quite easy to predict what the next 5 or 6 days hold for you. So, I hope you have a very enjoyable Nobel week here in Stockholm. Thank you very much.
Thank you.
Thank you.
Thank you.